Hi everyone. So in this video, I'm going to talk about some of the things we mentioned when we were discussing bacterial transcription and the initiation of transcription, the termination of transcription, and then how we move into translation. So the first thing I'll start off with is I'm going to just draw, there's one strand and the second strand of DNA in bacteria. And the first really big characteristic we see in bacteria is these boxes that we call them. So there's the -35 and the -10 box. And I'm also going to name the strands just so that it's easier. So that's 3', and then that one has to be anti-parallel. So then 5' and 3'. And so the way that we read DNA, so the way that RNA polymerase reads DNA is in the 3' to 5' direction. So it always reads this way, or on the bottom strand it would read this way. But what's really important is that the location of these -10 and -35 boxes within the promoter really set up which direction it's going to go in, right? Because if I didn't give you the promoter, technically the polymerase could either start on the bottom strand and move to the left, or on the top strand and move to the right. So really the idea is, okay, so how does polymerase know which strand to bind to and then which direction to go in? So really the big thing is we have this important factor called the sigma factor, which binds to these -10, -35 regions in the correct direction. So let's just say I draw this blob thing. Okay, so it's some sort of blob factor, the sigma factor, and it binds to the -10 and the -35 boxes within the promoter. What's really important about, I'm going to label it sigma factor, is the fact that where it binds, it could have bound on the bottom strand, but where it binds defines which way that transcription is going to go. Because at the end of the day, right, RNA polymerase is going to bind sigma factor, and that orients it towards going in the direction of the gene, right? Or else it would go kind of backwards, and it wouldn't really make sense. It would go upstream relative to the gene. So what's really important is the fact that the location of the -10 and the -35 boxes define which way sigma factor binds. Because as we discussed in class, sigma factor isn't a symmetrical molecule. So the way that it binds is really specific to the location of the -10 and -35 boxes. So now that it's bound, I'll draw another blob. So this is RNA polymerase, and it's going to bind to sigma factor. So sigma factor basically acts as a landing pad for RNA polymerase to come in and sort of understand that, okay, that's where I have to go, and that's going to direct me in the right direction so that I'm transcribing the gene. So it binds to sigma factor, it's in the correct place, and now it's going to start moving along the DNA. So let's say, I'm just going to put it in a random place, but in prokaryotes, the start of transcription occurs at the +1 site, right? It's the first place, the place where the first RNA base is added. So let's say if we zoomed into this sequence, right here, let's say in the 3' strand we had A, T, G, C, C, A, G, G, T, something like that. Just on a small portion of the DNA. So if I told you that this was the +1 site, then my mRNA would start with the complementary to that base. So it would start with C, and then it would go G, G, and then U, right, because it's RNA, not DNA, C, C, and then A, and then it would keep going, right, because this isn't actually that short. But the point is that the +1 site is where you would actually add the very first base. Okay, so now that we know sort of what's going on there, so let's say the RNA polymerase, it just starts going along the DNA, and it starts reading, and it starts making RNA. Eventually, it's going to reach the terminator sequence. So the terminator sequence will be transcribed, and then basically what happens is that once it's transcribed, the mRNA will form this like hairpin loop. So let's say this is like my growing strand of mRNA. I'm just going to draw it actually in simple forms. But yeah, this is my template strand. And then I have my RNA polymerase somewhere here, and then my mRNA is sort of going out. So once it reads the terminator sequence, the mRNA will actually form this like weird hairpin loop structure. And so that actually ends up getting the polymerase to fall off the strand. So it just basically, all you need to know is that this hairpin loop forms and that terminates transcription. Okay, and then so let's say now that we have our mRNA, so I'm going to draw it in red. So since it had to be made based on the template strand, which is this 3' to 5' strand, we know that it has to be anti-parallel because it's complementary to that strand. So we know it's a 5' to 3' strand, like the way that it was made. So this is our mRNA, and now we have all of these codons, right? We talked about the idea of a codon being specific to an anticodon, and that anticodon being having a tRNA that's carrying an amino acid. So for every codon that we see, there's a specific amino acid that corresponds to it. But one really important thing to note is that we know that there's a specific codon that corresponds to the start codon, and that's AUG. So let's say our AUG is somewhere here. So it's not actually at the very first base that was transcribed. And this will actually happen basically in every case because we know that in this region that won't be translated because it's upstream of this start codon, right? This is where we would actually start adding our very first amino acid. So we'd add methionine, and then we would add, right, we would add more as we go down the strand. But the important thing to note is that this region is untranslated. So it's the 5' UTR. And somewhere in here, there's a little sequence. Well, I don't know if it's little, but there's a sequence that's really important because it actually is recognized by the ribosome. So it's the ribosome binding site, or the RBS. And in prokaryotes, it's also equivalent to the Shine Delgarno. I might have spelled this wrong, but yeah, so we can really call them equivalently, but basically it's important to note that there's a specific sequence that's recognized by the ribosome, and it binds there, and then it moves in the correct direction based on where it's bound, basically. So that is, yeah, that's crucial to note, that we won't necessarily see the very first codon that you read isn't the first amino acid that's added, because the start codon is an AUG. So that's something that is important to keep in mind.